All right, hi guys. Welcome to another video of me. I'm in my living room. You can see the animals behind me, back there. And the dogs are literally right in front of me right now and staring at me. So if they start barking and it gets really loud, I apologize. All right, so this is your enzyme homework notes. So make sure you update your table of contents. We talked about enzymes today in class, and these are just a couple notes that we didn't have time to write down. This is what we did in class today in case you were not there. All right, so the function of enzymes, as we talked about, is to speed up chemical reactions in the body. So if you, for some reason, did not write that down, definitely write that down. But the reason that they can speed up reactions is because of this. Enzymes lower the energy that's needed to convert the reactants to the products. So reactants are what you start with. So say you start with lactose, which is the milk sugar, right? O-S-E, lact is milk. There are the dogs. Lactose is the milk sugar. So the enzyme lactase is going to break down that milk sugar into glucose so that you can digest it. So lactose would be the reactant. The product would be glucose, because that's what you finish with. So reactant is what you start with, product is what you end with. Well, enzymes, what they do is instead of taking this much energy, look at the black line, instead of taking this much energy to go from the beginning to the end, they lower it. They make it smaller, so it becomes easier to, get over the hump to get down to where you want to be. The amount of energy required to go from reactants to products is called the activation energy, which you definitely need to know. So enzymes lower it so that less energy is needed so everything can go faster. Here's a way to visualize it. So if you have a ball that you're trying to roll to the end of the hill, the ball is your reactant, what you start with, and normally in the body, if there were no enzymes, it would take this much energy. If you're following my cursor, you would have to go all the way up to the top of the hill and roll all the way down to get to the product. Notice you start in one place and end in another. But with an enzyme, the reactant, notice, starts in the same place, but the hill is much smaller because the energy required is much less. So the enzyme lowers the activation energy. You go over just a tiny hill, and you end up in the same place with the same product at the bottom. It just took you less time, and it was easier to get there. I can show you with this book if you want. So say you're trying to climb to the top of the book to get to the other side, right? So we're going up, 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 and over, and down. That's how it would happen normally. But with an enzyme, now you only have to go this far. So you go up the book, over, and down. Not as far to climb, easier to get to the other side. So here's a common test question you might see phrased one way or another. How do enzymes help the body function? So one answer, or any questions related to this answer, might be enzymes speed up reactions that would otherwise happen too slowly. So for example, when you eat food and enzymes digest that food, if you didn't have enzymes, that food would sit in your stomach. It would eventually break down over time, but it would happen so slowly that you would just get a brick in your stomach and you would die. So without enzymes, we would not be able to eat food. So that's how enzymes help, because they speed up reactions. The other answer that's related is that enzymes decrease the activation energy that's needed to catalyze a reaction. And to catalyze means to start or to speed up. So the answer either has to do with speeding up the reaction or, and, because they're related, lowering the activation energy. So you're welcome to write that down. It might help you on tests, but you don't have to. The other really important part that we briefly talked about in class is that enzymes follow a lock and key model. So here's the enzyme, this is the lock, and here's the key that fits perfectly. So enzymes follow a lock and key model where the enzyme binds to only a specific substrate. So if this substrate had actually been like a big square, it wouldn't have fit inside what's called the active site, or like the little place where you put the key, and so the enzyme would not have been able to work on it. So the enzyme, when something enters it that fits, it actually changes shape, so it goes from this to this, it like hugs it, and when it lets it go again, the substrate comes out, and it comes out as two separate things. So it comes out as the products. So it's called a lock and key because the lock can only be opened by one key, just like the enzyme can only be opened and closed by one substrate, and therefore can't change everything. Here's another example. So if the substrate was this shape plus this shape, so like a half oval plus a circle, it would fit because look, the enzyme has a little active site here where they would fit. Once the substrate enters, it'll kind of hug it, and then when it lets it go, it'll break it up, and now it's two different products. So why does lock and key matter? Because there are 75,000 enzymes in the human body. So can you imagine if they all worked on everything? We'd like be exploding because so many things would be happening. So enzymes only work on one thing and bypass everything else. So for example, proteases digest proteins and they will not work on carbs, and amylases will digest carbs and will not work on proteins. So do enzymes like all environments? No, they do not. They only work on their one substrate in their specific acidity, called pH, in their ideal temperature. So when asked, why are enzymes considered specific? You should know why. All right, good job, guys. See you soon.